Hi, my name is Mary Park. This is week 2, lab 3, and it's on bacterial enumerations, dilutions, and viable plate counts. So in the experiment, I had already prepared the series of bacterial solutions, and I already performed a viable plate count to determine the CFU/ml of a sample. So what I'm going to show you right now are the materials that I have used to prepare this experiment. What I have here are the tubes that I already had labeled. So I had to label six test tubes, 10 to the 1, 10 to the 2, 10 to the 3, 10 to the 5, and 10 to the 6. And then I had it to make sure I also had to label the agar according to what the tube is. So I had 10-1, 10-2, 10-3, 10-4, and 10-5. I also have, what I need to do this aseptic technique. So I have alcohol as well as the candle lighter, and what I need to make sure that I do this safely with the goggles, the mask, the gown, as well as gloves. So with this and preparing the dilutions, first of all, I had to make sure I mix the 1/4 of tablespoons of Saccharomyces cerevisiae from the yeast into a disposable cup, which I had already shown you earlier in the video. And after doing that, I would let it sit and then let it sit for 10 minutes. So after I had set things up, I had to add 2.25 ml of water into each of my tubes. So I had to do that, and then I would have to pipette 0.25 of the S. cerevisiae solution into the tube. So I would have to put 2.5 into 10-1, and then had to repeat the same step from that into 0.5 into 10-2, and then so forth. After that, I had to, the second part was to make sure that I had pipette 1.125, which is equal to 4 drops, into the S. cerevisiae solution of the test tube onto the agar surface, which was responding to the agar plate. So 10-1, 4 drops into the agar, and then I had to repeat it, same thing, for each agar. Then I had to, once I made sure that each had 4 drops, then I had to, corresponding to the tube, like I said, I had to incubate it in my pantry with the lid side up for 12 hours, to allow the liquid to absorb into the agar. And then after 12 hours, I would have to invert it to incubate for another 36 hours. And then after 48 hours, then it was a total incubation, and I had to count the number of colonies, which I will be showing that in my results in the next video. Thank you.